Yes,
All right.
looks good.
Okay.
I guess before we get started, are there any questions? And I'm sure Dr. Mwangi is going to ask you the same thing.
Right.
So again, you're going to be lecturing not only today, but also on Wednesday.
Wednesday, yes.
On Wednesday because of your jury duty.
The civic duties, yes, that we have to honor, yes.
Yes.
I was thinking of telling them, hey, I have a class, but of course they'll not take that.
Yeah, they won't. Oh, that's too
They
bad.
won't. Yeah.
And then next week we'll have Monday and Wednesday of presentations. We have student presentations on Monday, Wednesday of next week. Okay. And then there is also just to mention, don't forget that there's another assignment, peer review assignment that's going to be due here by Sunday. And if you have, be sure to look at it. And if you have any questions about it, be sure to ask me either by email or by when we meet again on Wednesday for class. Okay, everyone's pretty quiet. So with that, I guess I'll hand it over to you, Dr. Mwangi, if you want to get started. We were just talking about type 1 hypersensitivities, and now you're moving on to 2.
Correct. So we'll pick up from where we stopped talking about hypersensitivity reactions. And just to follow up on what Dr. Adam said is that we'll wrap up, we'll cover as much as we can today. So don't worry if we don't get finished. If we ever out to get some of the mucosal immunology covered today, that'll be fine. But if not, then we'll wrap up everything on Wednesday. Okay. So, and feel free to ask questions or send some questions so that we can answer them or address them as we progress. So, key information to remember: hypersensitivity reactions are recall responses. So there has to be a prior exposure. Okay, for type 2, they typically occur within hours as compared to the type 1. But what I want you to know here is that the key effector mechanism, the key driver is the complement. Complement has to be activated, and it's the primary effector mechanism of type 2 hypersensitivity. But what triggers those? What triggers the complement activation? The best 2 complement activating antibodies, IgMs and IgGs. Okay, and so then we'll see the outcome of what happens with regard now to tissue damage or the so-called lesions that are classical of type 2 hypersensitivity. So let's look at these types of antibody-mediated diseases. So first one, you have injury that is caused by anti-tissue antibody. So here, the antibody has to be there, or you have a memory antibody, and memory B cell that secretes this antibody upon re-exposure to the allergen or to the stimulant. And once those antibodies are triggered and then they bind, once those antibodies bind to the cognate target, the subsequent or the secondary reactions are the ones that causes damage. And in this case here, you're seeing that complement gets triggered, and then you have now the activation of neutrophils and macrophages, and the materials or the chemicals they release is what causes the tissue injury. So number one, tissue injury mediated by anti-tissue specific antibodies. So these antibodies that do typically cause these type of reactions are normally autoreactive antibodies. Okay, so they're mostly autoimmune mediated reaction against self antibodies. But it happens to be quite a number of these antibodies are actually raised as a result of what is termed as antigen mimicry. Okay, and it's quite it's common, especially in certain individuals that have got propensity actually to have these type 2 hypersensitivity reactions. So the best examples that we have here is these autoreactive antibodies that arise because of exposure to a pathogen, streptococcus or streptococcal infections, that end up now producing these antibodies directed against the pathogens. However, they not only take care of the pathogen or mediate the pathogen clearance, they end up having an ability to recognize certain epitopes. And in this case here, these antibodies end up recognizing epitopes on the heart or the heart tissue. And the subsequent outcome is what is termed as rheumatic fever. So inflammatory disease that can end up causing heart failure. So that is one example. The second example, so the first one here is direct trigger by the antibody itself. The second example here is now involvement of an immune complex, whereby now you have antigen, okay, that is recognized by existing or pre-existing antibodies. The immune complex is now the trigger here, okay, which is now different from what we just saw. But these immune complexes, they just don't end up causing problems everywhere. What happens is that when the blood and the fluids in the body keeps on circulating, and they end up now in small blood vessels or in small vessels in the body where they get now stuck. Okay, so because they're complexes, okay, and IgM is actually quite good at this because of its pentameric structure. So what happens is that the deposition within these blood vessels is what is now responsible for causing vasculitis. So antibody-antigen complexes results into complement activation, and then of course the ensuing immune reactions that now of course recruits other players such as neutrophils and are now causing inflammatory reactions that end up causing the damage to the walls of the vessels. Okay, so you can also have the same complexes ending up in specific organs. So the first one here, you're talking about vasculitis in vessels. Majority of cases will end up, a majority of these complexes will end up in the kidney because the body is attempting to get rid of them. Okay, so if they end up in the kidney, they end up now causing damage to the nephrons. And that is now what is now referred here as post-streptococcus nephritis. And under serious conditions, you can end up resulting in kidney failure. Okay, so how do these type 2 triggers, how do they mediate their mechanism? Let's look at a few examples of the effector mechanism. The first one is opsonization, whereby now you have a pre-existing antibody binding to a target cell. That's number one. And then the opsonized cell is taken up by a phagocyte. On the other hand, now you could also have the opsonized target cell triggering complement activation. And the complement activation in this case here, direct killing is not what happened. Is complement is not killing here directly. It is the generation of opsonin, of opsonizing complement byproduct, C3b, that now participate in opsonization of the target cell that results into this enhanced uptake of the opsonized cells by phagocytes. And that internalization of that cell results into the destruction of the cell within the phagocyte. So the best examples that you might have come across and maybe that you already know about are these autoimmune mediated anemia or IMHA. Okay, and the target here is, the target cell is erythrocytes. So you have massive destruction of erythrocytes. And that can also happen to other cells such as platelets that results into destruction of platelets in what is termed as thrombocytopenia. Okay, so how about a second mechanism that this takes place is whereby that initial trigger, okay, of complement activation when an antibody binds, the byproducts that comes from it that are actually pro-inflammatory, the C3A, C5A, we know that they are not only, they're very good at actually, these byproducts, they're very good at not only activating and recruiting a complement, but they end up now exacerbating that, mechanism. So in this case, now you have the byproducts from the complement activation, but binding of the antibody to an antigen via the FC-mediated receptor can end up resulting into activation of neutrophils through the FC ligation. Both ways results into activation of neutrophils, and it is the activated neutrophils that end up now secreting these pro-inflammatory molecules, such as the reactive oxygen species, elastase, and so forth. And if these are released at the site of activation, it is these molecules that end up now causing tissue injury. Okay, so two mechanisms that we've looked at. Okay, the third one is a type 2 hypersensitivity reaction that does not cause death or damage to the involved components. In this case here, you have involvement of antibodies that end up interfering with the normal biological function. And I have two examples here for you. The first one here is autoreactive antibodies that bind to the thyroid stimulating hormone receptor, or THS receptor. These antibodies, if they now bind to this receptor, they can ligate the receptors. And you require a minimum of 2 receptors to be ligated. And they end up now functioning like a hormone itself. In fact, some of these antibodies are even better than the antibody, but than the hormone itself. Why is that the case? Because their turnover is quite slow. Some of them don't even get off. Their own mechanisms is quite strong. They can actually bind and continue now that signaling, and they don't get off. And then you can now have these antibodies mediated stimulation in the absence of the hormone that results into hyperthyroidism or the Graves' disease. Now, it can actually even get serious than that, because once it's triggered, there is a memory, and every time that it binds, it exacerbates that reaction. But then these antibodies, if they are the IgGs, what happens? They can also get recycled or salvaged via what? The neonatal FC receptor. So you can see that they have this extended half-life that end up making the situation worse. Okay, so but you're not having a cell destruction here, but now you're having an outcome here that is unusual. The other example here is presence of antibodies that recognize the acetylcholine receptor, and which means that they can bind to the receptor and block the ability of the acetylcholine to actually mediate a biological function. And these antibodies end up now causing paralysis, and that outcome or the disease is myasthenia gravis, is the name given to that condition. Okay, so there are other causes of type 2 hypersensitivity, and in this case here, we are talking about the trigger and sustenance of such scenarios or such mechanism. The example I'm going to give you here is called autoimmune reactions caused by incompatibility between the donor and the recipient. So this is mainly caused by leakage of cells from the neonates, and that's why it has this term neonatal isoerythrolysis, and it affects humans, it affects cattle, it affects horses, and so forth and so forth. So the hemolytic disease of the newborn, I wanted to sort of give you a very good explanation, a very simple explanation. You're looking here at a neonate that has inherited traits from the father, okay? But the mother has never been exposed to that trait. So these traits are typically encoded on the Y chromosome. So the fetus here has now what? So you can see here, it's the boy's thing, right? Okay. So they're bringing this trait they acquired from the father. But if the cells leak during the birth, the mother gets exposed to those traits. The first time, no big deal. But they create that low level of memory. If that scenario is repeated again, there is a very strong recall. And so the mother starts now making antibodies against that marker, and quite a number of these markers, like the rhesus factor in blood, those antibodies from the mother will end up being dangerous, because if now the baby suckles and takes the colostrum that is now having antibodies against those markers on the erythrocytes, they're going to mediate destruction of the neonatal cells. That is a simple explanation here, okay? And so these fetal cells that I've talked about here is that that leakage at birth is the main trigger that and I'm now producing these outcome here. And as I said, is that the subsequent exposure is what really causes problems. Okay, and it's a serious problem, especially in humans. And of course, by the time it was discovered, there were certain strategies that came about on how to mitigate that, and we'll see some examples, some of those mechanisms at the end of this. Okay, the other example of a cause of type 2 hypersensitivity, this one here is termed secondary, because what happened here is that you have generation of altered epitopes or new epitopes being created by underlying causes. The best example here is folks who are allergic to drugs such as penicillin. And penicillin is such a small molecule, so it falls into that class of haptic drugs. So what happens in this case? A good example here is that these folks who are allergic to penicillin, penicillin tends now to form complexes that end up attaching or being present on the surface of red blood cells. Okay, and what happened is that distortion of the red blood cell surface can trigger now binding of the complement byproduct such as C3b, and you can envisage such as the alternate activation pathway. Now, the opsonized penicillin-coated RBC becomes a target of phagocyte. The best, of course, phagocyte here is a macrophage. It takes that complex, degrades it, and it has a very high chance of now generating peptides or epitopes, which compose of proteins or residues or components from the red blood cells membrane, fuse now to the penicillin. So the penicillin here being a haptic. So this epitope here can end up now being presented to CD4 T cells that end up inducing a Th2 or promoting development of a Th2 CD4 T cell response. It is these Th2 T cells that end up now providing help to B cells and generation of a plasma cell and a memory, such that every time now you have this person getting exposed to penicillin, they have that allergic reaction, because what happens is that you have those memory cells being triggered to secrete antibodies. Those antibodies now bind to the penicillin bound on the red blood cells, and those antibodies can trigger destruction of that red blood cells via the complement lysis. On the other hand, they're also very good at opsonizing the penicillin conjugate or penicillin coated RBCs. So then they get taken up and they get destroyed, leading into anemia. Okay, so another cause of, or another example of secondary immune mediated class 2 or type 2 hypersensitivity is generation of these altered epitopes on infected cells. And what happens is that infectious agents, especially hemoparasites that have a tendency to infect cells such as red blood cells, what happens is that they end up now creating these unique signatures or epitopes on the surface of these cells, which become a target of antibodies that then end up opsonizing those cells, and then those cells become targets of destruction. Best example here is this tick-borne agent called Mycoplasma canis, and also Mycoplasma feris has been demonstrated to mediate this secondary immune response that results into destruction of the affected cells, and hence you have anemia, or the affected animal or affected host becoming anemic. Okay, so These examples that are given here, I mean, they're just few, but there's a lot. There are many more examples that actually fall under this category. So, how do you mitigate this class 2 type 2 hypersensitivity? If it's hemolytic disease of a newborn, and you've already characterized that this mother has now high levels of antibodies against these markers, what you do is that we hold colostrum. So you can give that neonatal alternate colostrum from other unaffected donors. Or if it's a bit too late, you can end up now providing blood transfusion, but you can also now provide glucocorticoids if you have exposure of that neonate taken in, let's say, colostrum by mistake. So the other mechanism here that, or the other mitigation strategy is to determine whether the causative mechanism is primary or secondary. If it's secondary, meaning that you have an infectious agent being involved, so you can treat if there is a treatment option, okay? Or if it's drug related, for example, in the case of penicillin, then of course you mark that individual. Folks who are penicillin resistant, of course, they know that they cannot take penicillin based drugs. So if it's primary disease, what happens is that you can use immunosuppressive drugs such as glucocorticoids, or you can use other drugs such as thiopurine, which inhibits IL-2, and can end up now block proliferation of T cells, mostly CD4s. Okay, so those are the treatment options that you have, and that brings us now to the end of type 2 hypersensitivity reactions and a few examples of mitigation strategies. So if there's a question, I can handle it right now, or if not, you can actually send that and we address it. But if not, we can switch gears here and talk about type 3 hypersensitivity reactions.
So are there any questions to follow up? There are lots of different, it seems like there's lots of different scenarios of type 2, but as long as you're remembering IgG, IgM, and complement seem to be the most important commonalities for those different types of diseases. Is that true?
Yes, that's true. And in fact, what you've said here now is in here, and we need to sort of make that clear that the involvement of immune complexes in type 2, you don't have to confuse that now with the involvement of immune complexes in type 3. Because in type 3 now, you can see you have an IgG mostly, and then you also have a role of IgA in certain circumstances. But the main effector mechanism here is what? It is the neutrophils. Whereas in the type 2, the main driver was what? The complement. All right. Again, you can see here that the time frame is actually similar. It's actually within hours if there's prior sensitization. So in this case here, the generation of these immune complexes, when they get now deposited in tissues, they end up now triggering tissue injuries. And so if you can look at some of these mechanisms, which occurs within these vessels. And the main affected tissues are going to be the skin, the lungs, the eye, the kidney, quite a number of these organs. But again, these events or these type 3 hypersensitivities can occur in multiple organs or multiple tissues concurrently. But it follows distinct phases. So there are 3 phases. The first one here, there has to be pre-existing antibodies or pre-existing immune response. So once an antigen is introduced, you have formation of immune complexes, and then those immune complexes get deposited in tissues or the blood vessels. The second step here is that you have a role of the complement, but the complement here is actually not the effector mechanism. It is an enabler in the sense that it gets activated, and the complement byproduct, the pro-inflammatory byproduct, the C3A, C5A, they get released. And it's these molecules that play a role now in recruiting mainly the neutrophils And to a lesser degree, you also have activation of mast cells, and you're going to see an example of that. The third step is that you have these activated neutrophils and mast cells being triggered or being they end up now undergoing degranulation to release their contents or enzyme contents and other factors such as the reactive oxygen species. And it's these released chemicals that end up now causing damage to the tissue, to the surrounding tissue. So let's look at this in terms of a timeline whereby now you're talking of injection, let's say, of an antigen locally, let's say within the skin, okay? That antigen gets recognized by pre-existing antibodies. And the second step is that you have formation of a complex. Is this the complex here now that triggers complement activation? And the complement byproducts comes into play and start driving this process. So by the way, C5a, besides now activating the neutrophils and so forth, is also a very good stimulant of mast cells. And you know, within the tissue, you're going to be having these resident mast cells. And once they get triggered, they start releasing what? Start releasing their contents, such as histamine and other factors. That those molecules, when they get triggered, they are able now to activate the endothelial cell or the epithelial cells within the surrounding, and that results into the leakage or leakage of cells into the affected tissues. In this case here now you're having a combination of factors. The complement byproduct, which are functioning here as chemoattractants for neutrophils. They are also not triggering the histamines released from the mast cells and other factors that causes this tight lining of the blood vessels to have this formation of these gaps that allows now these cells to migrate to the tissues. The activated neutrophils then gets triggered now to release its content, and that's what now causes the damage. So there are several examples. Okay, and these examples, and for type 3, specific route of antigen entry matters. So if you have subcutaneous entry, you have now the resulting disease which is called adverse reaction, and this mostly affects the perivascular area. So if you have, so you can imagine that the first case here is mostly going to be very localized, all right? But on the other hand, if you have intravenous entry of this, of the allergen or the trigger, if now you have antigen antibody complexing within the blood vessels, you have vasculitis. If you have those deposits being ended up in the kidney, you end up having nephritis. And if they get deposited within the joints, you have arthritis. So you might now consider that the intravenous entry of this allergen or the trigger is going to have a systemic effect. But within that context, you can see that it's affecting specific organs or tissues, whereas low dose subcutaneous entry is actually localized reaction. Okay, so examples of this. Horses get this pathogen called Streptococcus equi. First time, no problem, but once the memory is there, re-exposure is what results into Streptococcus equis vascularity lesion as seen over here. That end up now reacting or occurring in a very specific mechanism that we've just talked about. You could also now have deposition of these complexes within the joints that end up causing rheumatoid arthritis, okay? And you can also have this scenario whereby antibodies directed against DNA and nucleoproteins and other nuclear components end up causing systemic lupus erythematosus. And of course, it's called systemic because it affects multiple organs, such that you might end up having the kidneys being involved. You might have this reaction within blood vessels, and also happening in joints that results in arthritis. Okay, so few examples of type 3 hypersensitivity reaction, but the question is, how do you manage this? So if a pathogen is involved, which rarely are you going to capture that? Because by the time maybe it's flaring up, the immune system has already taken care of the pathogen. Okay, so if that's the case and you have an active infection, yes, one of the things that you could do is treat it. But as I said, typically by the time that is coming up, it's way past that stage. So you need to look at other mechanisms for managing this. And so you suppress the immune system by use of glucocorticoids, cyclophosphamide, and so on, to be able now to dampen, or to be able now to taper down the uncontrolled or out of control immune response. Okay, so type 3 hypersensitivity reactions. Very, very brief, and as I said in the first lecture here, is that it is an injustice actually to cover these hypersensitivity reactions the way we do, but of course, we have to have a high-level overview. Typically, there's more to it than what we've covered. But again, we're hitting the main points. So, any questions for that, or we can switch to type 4 hypersensitivity reaction.
Any questions? I know it's a lot, but I think it'll make more sense after you review it. Any questions? No questions?
Okay, and so I hope that you're able now to differentiate between the immune complex involvement in type 2 and immune complex involvement in type 3. Regarding type 4, you hardly have any antibody contributing to this, but in some cases it happens. But they are secondary players. They are triggered once you have a cellular immune response kicking in as a type 4 hypersensitivity reaction, because here, the main players are, the keyword here, activated Th1, activated CTLs, activated macrophages. And because it builds up very slowly, that's why it's called what? Delayed hypersensitivity. All right? And so the outcome here is that you have tissue damage, cell damage, and so forth. We're going to see some of the examples of what happens. So this is exactly what we talked about. Activated Th1, CD4 T cell, CD8, that once they're triggered, and they're triggered by pro-inflammatory cytokines, they themselves end up now contributing to that reaction via release of several factors such as interferon gamma, TNF alpha are involved in this, that end up now creating a microenvironment that you have highly activated macrophages and several T cells, which end up now being involved in type 4 hypersensitivity. And as I said, it's delayed hypersensitivity. It's a delayed reaction because you end up now seeing it manifesting very well within a window of about 2 to 3 days, and the outcome here is some of the outcome here are actually obvious, such as the swelling, formation of granulomas, and accumulation of lymphocytes at the site of reaction. Or you could end up now having necrosis that is mediated by destruction of the target cells by CTLs. So I think this information that I've just mentioned here can actually be presented in another way and broken down into 2 parts. Type 1, or part 1, type 4 hypersensitivity reactions that are driven or mediated by cytokines, inflammation. So in which case now you have pre-existing memory of CD4 T cells that are directed against the causative agent. And of course, they end up now recognizing an antigen presented in the context of the class 2. And CD4 T cells are actually very notorious. If they get the right signal, they proliferate very, very quickly. And they also release massive amounts of cytokines. And it is these cytokines that now end up becoming the fuel or the flame that is needed or that is involved in triggering phagocytes, the macrophages and the neutrophils. And once they're triggered or when they're stimulated, they end up now releasing these tissue and cell damaging molecules, such as reactive oxygen species and other enzymes. The second example here is T-cell mediated killing in a classical effector to target, where you're having a memory CD8 T cell being triggered to get activated and expand because it's recognizing a cognate epitope presented by MHC class 1 by a host cell. And in that case, the CTL, once it's triggered, it does the job, which it's well trained to do, and that is actually now causing death of that target cell. So let's walk through these step by step, and first of all, look at how you get the CD4s to get involved. So antigen introduction into the tissue results into the uptake of that antigen by AP6, antigen-presenting cells. Then they generate peptides that are presented on the surface, and since there's already pre-existing memory, that subset of the T cells is going to be attracted to this site here and recognize those epitopes. And within a window of about now 3 days, they proliferate. And of course, when they're proliferating, they reach a certain a number whereby, of course, they're not only proliferating, they're secreting what? Cytokines and chemokines. And because of accumulation or the reaction that is happening over there, you can see that there's going to be fluid accumulation, and also you're going to have injection site swelling because of all those activities that are happening there. Depending on the allergen, okay, you can also even touch the affected and you can feel that it's hot. Okay, so that depiction here really gives you a simple presentation of the events that takes place in that classical CD4 T-cell mediated type 4 hypersensitivity. Now, that type 4 hypersensitivity reaction for a long time was exploited as a diagnostic tool for detection of folks who have had prior exposure to TB. And it was a simple test that is termed as tuberculin skin reaction, whereby they utilize Mycobacteria tuberculosis extract. Okay, and what happens is that this extract called PPD, it's injected into the skin. Okay, you would go now into the clinic or to the hospital, they inject it to you and tell you go home and come back after 3 days. So after 3 days, of course, they're going to have one spot where they injected, let's say, PBS, and spot now that they end up injecting PPD. So after 3 days, if that person has had prior exposure or they're having an active TB, which means that they have memory CD4 T cells, there's going to be a tubercle or a glioma formation at the site where the PPD was injected. So that was old technology. I think is still being used in some places, okay? And the problem here is, if you get tested once and they inject PPD to you, what happens? And I'm assuming you are negative, but now you've had a dose of PPD. Yeah. What does that mean? You can never have that test again because you already now start making what? Yeah. Immune response against PPD. But you never got exposed to the pathogen itself. Yeah. Do you see that the limitation for that? Yeah. It's only a one-time thing. It's a one-time thing. All right. Now that assay or that test is still done here, but now in cattle, because TB, Mycobacteria infection in cattle is prevalent, especially in certain regions in the United States. So, and there is a need to actually identify the affected animal and be able to cull it. So they have this PPD and they inject it into the animals. Within 3 days, they come back and palpate the site, and sometimes if the reaction is massive, you can actually just see it. they're able now to eliminate those animals. Now, that was sort of, again, an old technology. These days with PCR and other advanced techniques, you can actually now check. Although sometimes it becomes very complicated because you could have this bacteria hiding where? In phagocytes within the lung. And if you don't want to kill the animal, so you're looking for the pathogen in the periphery, you might not detect it, but the animal has it. Okay, you should see that. Okay, another example here is of our type 4 is the famous contact dermatitis. Maybe some of you have had an exposure to poison ivy. If you have, by the time now you're getting this kind of reaction, know that that is not the first time that you got exposed to poison ivy. So poison ivy has actually been shown to act as a hapten and stimulate generation of CTLs that are actually specific to certain determinants on the keratinocytes. And so upon re-exposure, triggers that, and then you have CTLs that end up now destroying the keratinocytes. So you end up having an affected person, like this hand you're seeing over here. You wouldn't know that it's the CTLs that are actually driving it. Now, the actual mechanism of how this happened, the details is not quite clear. All right. So other examples, multiple sclerosis in humans, and this might be something that you might be familiar with, but again, what happens here is this is as a result of destruction of the myelin basic protein. Okay, that insulation that is around the nerves, and all that, or even the brain that get destroyed. The notorious trigger for that is Epstein-Barr virus. Because Epstein-Barr virus, or EBV, its DNA polymerase has an epitope that mimics or looks very, very similar to certain determinants on this myelin basic protein, such that exposure to EBV triggers very nice CT response against that epitope, but they are autoreactive. They can end up now destroying this insulation around the nerves or the oligodendrocytes. Okay. Second, another example here is this phenomenon called polymyositis, which is a progressive muscle destruction that is driven by cellular immune responses that results into degeneration. Okay, and it's quite it's characterized by accumulation of lymphocytes and macrophages that end up infiltrating the muscle. Diabetes, okay, classical destruction of the cells that produce insulin is something that you're familiar with. And again, the main trigger here, or the most effective mechanism here, involves T cells destroying those cells. But then once the T cells do the job, the debris that arise from there can end up now producing epitopes or antigens that trigger B-cell responses and generation of antibodies that also contribute to that process. The last one here is the lymphocytic thyroiditis, which is also CTL mediated destruction of the thyroid epithelium. Okay, and quite a number of canine breeds. So the beagles, the beagles and the Great Danes have been well documented as being predisposed to lymphocytic thyroiditis. Okay, and so the trigger here again is this type 4 hypersensitivity reaction that results in development or generation of CTLs that end up now destroying the thyroid epithelium. Okay, and the resulting outcome is thyroiditis. All right, so what are the treatment options for type 4? So if it's a chemical and or another substance like, of course, as poison ivy, you avoid it. Okay, but in the majority of cases, once it's triggered, you can actually use anti-inflammatory drugs, glucocorticoids, and if it's if infection or infectious agent is involved, then you can be able to treat that. For example, in the case of mycobacterium infection. So with that, you have your learning outcomes here that in brief, gives a highlight of what we've covered with regard now to type 1, type 2, all the way to type 4 hypersensitivity reactions. But so I'll pause here and give you a chance to ask some questions, see whether we can be able to now to provide further details if need be.
Any questions? And then I just put a suggestion in the chat box. It's nice that you have that nice table. What you might also consider is expanding that table to provide examples of some real examples as discussed, and then also treatments for those different disease types under each hypersensitivity. That's just a suggestion for studying purposes. Any questions or comments or need for clarification?
Okay. If none, let's switch gears here and talk about mucous immunity. Okay. And particularly bias our focus on immune responses to intestinal parasites and see what happens there. So here you have your list of your learning objectives, and you're going to go through all these 5 objectives and cover a bit of this in detail. So the immune system is split into 2 components. The peripheral immune system is unique and separate than the mucosal immune system. The mucosal system seems to be separate, such that if you immunize peripherally, you hardly induce or you hardly have effective induction of immune responses within the mucus. So if you really want to get a good vaccine that works against microbial pathogens that infect via the mucosal route, you better come up with a strategy of actually eliciting immune response at the mucosal level. Okay, so you can see here, for example, the information. This slide here, you can see that if you take the dog, for example, and this could also even be a human, you can see that you have a huge network of these lymphoid organs, the lymph nodes, especially within the skin and other tissues, such that each and every part of our bodies, you have a lymph node that serves that, the so-called draining lymph node. Okay, so that is now within the periphery. Now you can envisage that within the mucosal surface, you have something similar. Okay, but before we got there, I just want to give you an overview of the other important immune mechanisms that are important on the body surfaces, and they're listed here, and here we're using the dog as an example. Again, combining the largest or the primary barrier, the innate barrier, of course, the skin, an intact skin. And then when you start now looking at other factors such as the tears, you can look at the mucus within the the gut and so forth. And we're going to go into those. But I wanted now to let you know that yes, although now you're looking at an animal like a dog here, or if you now even substitute and talk of human, it's not now these factors that you're talking about by themselves here that you have this huge population of normal flora, all the good bacteria. And in fact, if you look at a human, you have more bacteria than human cells. So are we bacterial or are we something else? Because the number of bacteria we have at any time outnumber the human cells. All right. Now, we can't cover all these, but we just wanted to focus a little bit and talk a little bit about the immune components that are critical for offering protection at the mucosal surface, the so-called mucosa-associated lymphoid tissues, or the mold. So this mold mass is so huge that taken together, the number of cells you have within these lymphoid tissues, the mold, outnumbers, it exceeds all the cells in the lymphoid, the peripheral lymphoid cells that I refer to, the exterior, the secondary lymph nodes, the spleens, the bone marrow, and so forth. So you can actually now appreciate the massive deployment of these potential infectors. But the question here is, why is that the case for the mucosa? Any thoughts? Simply for living creatures like us, if you're going to get exposed or infected, what is the likely route of infection? The portals of pathogen entry are obvious, right? The eye, the nose, the mouth, and so forth, right? Okay. And then the fact that unlike the skin, an intact skin can really take care of things, But if something gets into your nose, for example, if you're next to a person with COVID, you're going to get it very easily because the lining, the epithelial lining on your mucosa, it's very, very vulnerable. I mean, it's very easy to penetrate. It's very easy for pathogen to get there. So the investment or the availing of this massive number of cells at those portals of potential pathogen entry it doesn't make sense. Okay, so let's look at some of this. Okay, so the first one here is the nasal associated lymphoid tissue, or the node. Okay, and that one takes care of very early events that happens within the mouth. So the tonsils is one of them, and this is something obvious. Sometimes that what happens is that all of a sudden you have this inflamed tonsils here. You know, something is happening, right? Okay. So the other one is gut-associated lymphoid tissue, the gut, and we're going to be focusing a little bit more on the patch and see what happens there. Then you have the bronchus-associated lymphoid tissue of the ball. I'm just going to mention a few of that, but you're not going to go into details there. So we want to go to the mucosal or to the gut, because the epithelial barrier within the gut, it's really quite interesting, but more importantly, it's quite complex. Now here we have this slide here. It's actually giving you this tissue of a healthy mucosa, very healthy, nice looking, and when you look at it from this stain here, you might underappreciate the complexity of the components, the immune components that are at play here. To do that, we have to get to this slide. Okay, so looked at this from this slide, you can actually now divide this the epithelial, that region into four: the intestinal lumen, the mucosal epithelium, the lamina propria, and then the mesenteric. So to get started, let's look at what you can call low-hanging fruits. Some of the most effective tools that are at play in terms of fighting microbial pathogens within the lumen. Massive number of the good bacteria, the commensal bacteria, which outnumber the bad guys, outcompete the bad guys. In addition to that, you have presence of IgA, majority of the species. We'll talk a little bit about IgA later. But then, before we leave the lumen, we have to appreciate that a healthy gut cannot be complete if you don't have good mucus and plenty of it. And why is that important? There's quite a lot of people who don't make enough mucus, and they suffer. They suffer. They have a situation whereby inflammation can get triggered by even minor things. So good mucus really is a key component in directing or mediating immune protection within this site. So when we come now to the mucosal epithelium, I want to start all the way from this end, which I'm using my cursor here to see, and walk through this step by step. So this mucosal epithelium, it's tightly packed, healthy layer of epithelial cells. Okay, but within that epithelium, you have different effector cells, and the first one here is intraepithelial lymphocytes, which I'm assuming that you're already aware about. Intraepithelial cell is the only immobile T cell, but it plays a very, very significant role within the gut. And how do we know that? That in the face of an infection, the number shoots up. And once it's resolved, it comes down. they're well poised, well positioned to be able now to mediate the effect because they have receptors that can be able now to bind to the target pathogen and be able now to trigger their destruction. So next to that, you have identity cell subset that is strategically positioned and then within or close to the mucosal epithelium. And then it has these dendrites that go through the gaps between the epithelial cells, and the dendrites can sample the activities that are happening within the lumen. Again, you're going to see how they play a role in mediating immune responses to that. Next to that, now we have the so-called the crypt, whereby it's rich in and endowed with these cell subset called the Paneth cells that are the job is to screen antimicrobial defenses. And these are highly charged peptides that in certain species, for example, in young animals, especially cows, they make a different variety of these defenses. And they're so potent because they end up obsolescing the pathogen and triggering killing of the pathogen. So next to that, we come now to some of the interesting cells that you have within the mucosa. The famous one here is the M cell. So why is it called M cell? Okay, because it has this trait of forming what are called, so the M cell comes from a microfold. So it stands for microfold. It forms microfolds that end up forming vacuoles that are involved in transport of larger molecules or larger components from the lumen into the pia patches, and we're going to study that a little bit more in a short while. And then next to that, you have now the source of the mucus that you talked about, the goblet cell. And again, you know, a healthy gut cannot be complete without this. So by now jumping into the lamina propria, this is an area that is actually rich and endowed with the lymphocyte, T cells, the B cells, and other factors or other effectors such as the mast cells, macrophages, and so forth. But more importantly, you don't only have them those cells there as I've listed, but you also have now memory B cells, memory T cells, ready now to go into action. And in this case here, you're seeing an example given here of a plasma cell, which is what? An antibody factory. And in this regard here, its job is to secrete IgA, and the IgA, we're going to see a mechanism by which now it's transported into the lumen to be able now to mediate its effector functions. Within the lumen, you also have this rich network of lymphatics. And these lymphatics are the ones which are now draining the epithelium. So you're having this constant flow of fluids from the epithelium. And those fluids are coming with all sorts of stuff that is coming from the gut. It's how some of the low molecular weight soluble antigens are actually transported from the epithelium to the major lymph node that you have in an animal. The mesenteric lymph node is the largest lymph node. And what happens is that it serves this gut here by now through this network of the lymphatics. And besides other soluble factors, that the soluble molecules that are being collected and drained into the mesenteric lymph node, you also have dendritic cells acquiring the antigens from the mesenteric and then transporting the cargo to the mesenteric lymph node, where now you have priming and expansion of antigen-specific T cells and B cells, and we're going to look at that in a few minutes. All right, so to look a little bit or to summarize that, then I can put it this way: that the mucosal immune system, you know, the mold here has large lymphoid areas, and again, you appreciate what I told you previously or why that is important. And of course, besides now continued exposure to microbial organisms, that is the route by which our bodies get exposed to the food we eat. And you're going to see some reason. We are going to discuss in brief why our bodies don't, in a majority of us, we don't react to food antigens, okay? So, and again, just to summarize here is that mucous membrane is the largest organ. And I've already mentioned that it's separate from the peripheral immune system. And of course, you have the epithelial barrier that is a first line of protection. And you've seen that I've mentioned here that you have production of antigen-specific memory B and T cell responses that are generated within these sites. And I'm going to show you or talk to you a little bit more of the location where they're produced and where they exactly their effector function. And lastly, we are going to talk about how the mold is involved in providing tolerance to the food we eat. So before we do that, let's just go through the epithelial barrier in brief and look a little bit more in detail these components that constitute these barriers. So the epithelial tight junctions, okay, they're important because if they're not tight, then you have trouble. Stuff can leak in. Okay, and so what keeps these together? So besides a healthy tight monolayer, you have this molecule glycocalyx, which essentially is a component of the mucus. Okay, and it's formed through combination, or it's a combination of several components involving the mucin, which are basically carbohydrates. It also lipid rich, and it has a bicarbonate free. So, and of course, you mentioned that it's secreted by goblet cells and so forth. So, the important information here to appreciate is the fact that this combination of these components allow nutrients to pass through after digestion, but not letting big micromolecules, especially pathogens, to just have a free entry, okay? And again, it's within these tissues that we talked about, the generation or the secretion or release of defensins secreted by the pathogen cells. And without repeating myself again, I think I gave you a good explanation as to their role in addition to what you already know. Last but not least, these epithelial cells are not just mechanical. They're not only now performing the functions that we talked about, they are also a great source of other factors that actually contribute in immune response, which include secretion of cytokines and chemokines. The examples given here is 10 TGF-beta, and their role is mediated immunosuppression, and we're going to see why that is important. But you also have cells, these epithelial cells releasing pro-inflammatory cytokines such as TNF alpha, okay? And that is important because if you have to have a productive induction of productive immune response within the gut, an immunosuppressive environment is not going to allow it. There has to be a mechanism, or there has to be a way of inducing an immune response, even if it takes a transient event to be able to induce an immune response towards an invader, so that now you can have an immune response that is going to be involved in eliminating that potential threat. Okay, so having said that, I want now to switch gears and talk a little bit about two key sites within the gut that you need to know about. The first one here is inductive sites. In other words, within this mold, you have sites where you have induction of antigen-specific T cells and B cells, and it's within these sites that you also have antibody class switching and so forth. Once they are generated, they are utilized, or they mediate their effector functions at sites referred to effector sites, which are a distance away from the inductive sites. It is where the induced effectors end up now playing a role in taking care of pathogens or killing infected cells. So, example of these inactive sites, the first one that we saw is the tonsils, which is present in most mammals except the rodents. So, meaning that exposure within the mouth region and the nose region, any pathogen invading or getting through there gets trafficked to the tonsils, and it's the tonsils now that is able now to mount an immune response to be able now to counter that. The other example is the pharynx, the nasal lymphoid tissue, the bolt, and the appendix. All those are inactive sites. But last but not least is what? The Peyer's patch, okay, which is a very active, immunologically active site within the gut. Now, what I need to mention here is that some of these sites are actually endowed by the rich in stem cells that actually repopulate the cells involved in this immune response. And I'm going to get to that shortly when I talk about IgA inducing antibodies. So, but to be able now to appreciate the events that takes place at the inductive sites, we have to ask the question, how do the antigens get there? Because we talked about all these barriers and all these pre-existing mechanism to block or to inhibit easy entry by the pathogen. How do antigens get into the inductive cells? That's now how we come to the M cells, the microphore cell. So it is strategically located within the epithelium, and it has this capability of now forming these microfolds or pockets that then that contain the content of microbial pathogens from the lumen, and then transport them across. Now, just below the M cell, strategically located are dendritic cells, the most effective what? APC. And of course, once the cargo has reached this end over here, the dendritic cell takes over. Now, within the intestinal mucosa, there are several dendritic cell subsets, and they have unique biological functions. Here, we're just going to talk about two of those. And the first one here is this one here that has this signature. It has a unique surface marker called CD11B. It has a chemokine receptor, CX3CR1, but that is generic because it's also present in other disease. Now, this dendritic cell is strategically located below the epithelium, and then it has the dendrite. This is the one that I was telling you that it's sampling the lumen. This dendrite here, how does it know what to capture and what not to capture? Because it has what? Pathogen recognition receptors. Those are the signatures it recognizes, and it has other antigen capture mechanism. And then those antigens are transported into, they're transported and processed and presented to T cells for induction of antigen-specific immune responses. So you might now argue or consider that CD11B positive dendritic cells within the intestinal mucosa, they are involved in priming antigen-specific or pathogen-specific immune responses. So they end up being, or you can actually categorize them as key players in creating inflammatory and conditions that are ripe for productive induction of T cells directly against microbial pathogen. On the flip side, you have this other dendritic cell subset, which is CD103 positive. You can see here that it doesn't have the CD11 marker. This dendritic cell, unlike the first one here, it does not have a dendrite that samples the lumen. What it does is that it waits for substances to make their way through the gaps between the epithelial cell. And these substances that make their way through this gap here are actually mostly the broken down food that we eat. Whereas the first one here is actually sampling microbial pathogen.
So the second DC subset here is endowed with an ability to make TGF better. It also releases electronic acid and a combination of other factors that are conducive for priming and expansion of T regulatory cells. And that's how the food we eat and other molecules or other antigens are able now to be taken up by this disease and create a microenvironment that is anti-inflammatory. Those other molecules besides now the food we eat, they fall under that category because they lack pumps. They lack pathogen-associated molecular markers. And that's why you can see that these two disease subsets, one is specialized in responding or in driving immune response towards anti-inflammation, where the other one is well endowed with the capability to induce an immune response geared towards generation of effectors for elimination of microbial pathogen. So to appreciate what I'm just going through here is that we want to look at a closer look at this, the discrete bio patches and see just below now the M cell, that within the past patch, you can see that it's rich in T cells, CD4 T cells, CD8 T cells. And in addition to that, you also have B cells. Now, you can see that strategically, once the antigen comes in, taken up by the T cells, degraded, and others, you end up now having priming of what CD4 T cells, and concurrently that antigen makes its way and gets recognized by what? The B cells. But there's an interface in between there whereby now the primed CD4 T cells get attracted now to closer now to or towards the B cells, and then I'm now providing help to the B cells for class switching and so forth. So just to remind you, the details we've talked about here about the detailed components of the gut, and of course the antigen uptake mechanism. The reason why I'm showing you here is so that now I can bring you to the focus here to really ask the question: how then do you have induction of antigen specific responses, specifically generation of different T cell subsets? There is or there are key determinants that drive this. So the M cell does recognize and uptake the pathogen, hands that over to the T cells. From that point onwards, the outcome is going to be driven by several factors. If it's a food antigen, you have a propensity, or you have a tendency now for the DC to present the antigen to T cell subset in a microenvironment, or in a microenvironment that it's going to be having high levels of, or preferentially high levels of interleukin 10 and TGF beta that ends up now skewing towards a Treg outcome. And the reason why this is the case is because food-derived antigens lack what? Pumps. They lack pumps. And that's why now you have this tendency now to generate Tregs as a default mechanism. And that's why we don't react to most of the food we eat, or majority of us don't at all. On the other hand, if the antigen being taken up by the cell has a pump that is unique to it, that is consistent with what you might, that is consistent with the what we know so far regarding characteristic of antigen. And in this case here, that it's going to be having Th2 type signatures. It ends up now promoting priming and expansion of the Th2 T cell responses. And most of these antigens are actually going to be derived from parasite. And so you end up having induction of a Th2 subset population that end up secreting these cytokine combination. Similarly, you also have antigens, especially intracellular pathogens that have got signatures that drive Th1 immune response with an overwhelming, prevailing interferon gamma secretion signature. But last but not least, here you also have antigens that have got pumps that are uniquely unique for driving a Th17 immune response, and ended up now creating a scenario whereby you're generating Th17 effectors that are actually capable of getting rid of endogenous fraud. So you can see from the events described here on how the M cell is acquiring the cargo or the target antigen, you can end up now with all these potential outcomes, mostly driven by what? Presence or absence of pathogen associated molecular patterns. Okay, all right. So having said that, I just now wanted to bring it together and tell you that the events that we talked about that are getting initiated through the antigen acquisition by the F cells, we saw the events that are taking place in the part. Concurrently, you also have antigens that are being acquired by the cells or by transported by the lymphatics that end up now in the mesenteric lymph node. So taken together, you are going to consider this is as what? The inductive sites. What happens is that those induced effectors, the B cells, the T cells, they just don't assert their effector functions right there. There is a rule, a simple rule. What happens is that they get transported by the lymphatic system, emptied into the blood system, and through the blood system, through the blood circulation, then they're distributed back into the body. But what happened here is that they circulate and go back to the gut because they have what is termed as homing markers. Once they get back to the gut, because of those homing markers, they're able now to enter into the effector sites. And it's there now that you have memory cells, memory B cells, memory T cells, asserting their function. Okay, so having said that, we just want now to look briefly at one of the effector molecules. A simple one here being secreted IgA, which is the main mucosal immunoglobulin in most species, except in cattle, where IgG is the most important. So IgA within the gut can be induced in two ways: T cell dependent class switching in the gut, and the other one is T cell independent. So I think we're going to finish this one here and stop here because we're getting to 5:20, and then we are going to wrap up this very easily by Wednesday. So regarding T cell dependent, what events do take place here? Back again to antigen acquisition by the itself. You have the cell antigen uptake and processing and priming of CD4 T cells, so that once they become activated, they provide now help to the B cell or the cognate B cells, which will have already also recognized the same antigen. And this event or these events within the Pius patch are unique. They're unique as compared now to, let's say, T cell priming, let's say in the spleen or in the peripheral lymph node. Because what happens here, you have a DC subset here that end up now secreting unique factors such as the TGF-beta and nitric oxide. You might be surprised, that some of these molecules are actually multifunctional. End result is that the events end up not influencing these B cells to switch from making IgMs to start making IgAs. And of course, you now have generation of plasma cells that are programmed to secrete IgAs. So they exit the inductive site through the circulation, then they home back into the gut, where they become resident and become factories for IgA secretion. And the IgA that is secreted from here then has to be transported into the lumen for it to be able now to mediate its function. Think that is quite clear. We'll stop over here, then we pick it up on Wednesday, we wrap up on the T-cell independent IgA class switching. Maybe I'll pinch 1 or 2 of your minutes to ask whether there is any question that I can answer.
Any questions? One that comes up often, and maybe you can, maybe this will come up later, but what about the commensal bacteria that are inside the gut? How does the immune system know that not to kill the commensals, but kill the bad guys? How do they distinguish?
Good point and good question. But these commensal bacteria, where are they? Where are they located?
Intestines.
In the lumen, right? They better stay there. Because if they don't, what of sepsis, but if you get now damage to that barrier and they get in, they're no longer good guys. No, they're good guys so long as they stay where? In the gut. They get it inside, they're going to cause problems. Now, so a healthy gut really restricts their stay within the gut. Now, you have dead bacteria, molecules that are being shed from these bacteria that can trigger immune response, right? Why doesn't that happen or minimally happens in a healthy individual? Why? Because commensal bacteria lack what? Pathogen associated molecular patterns.
They lack them?
So, good example. Bacteria that cause gram-negative that have what the pathogens, the bacteria within the gut don't make such ligands or such pumps like LPS. If they did, they would actually trigger the immune response right away. And you can actually do that experiment. Take a good bacteria, transform it with a plasmid expressing LPS, or make it transform that bacteria so that it can make LPS, and then you put it back there. It's not going to be a good guy. The immune response can't ignore it. Why? Because the immune system is pre-programmed to recognize what? Pathogen associated molecular pattern. Yeah. So, yeah.
Makes good sense to me. Any follow-up questions or other clarification?
Same thing. Those bacteria, they don't like classical flagella and whatever, because flagella is a right. You can make those bacteria express and of course you put it back over there, triggers immune response. Okay. All right. Any more questions?
Yeah, I have another question.
Okay.
So the bacteria, it doesn't have to be bad to cause a disease. Sometimes just having literally too much of them or too less of them and causing conditions. And how our immune system can know these bacteria are not actually bad, they're just not enough of these bacteria, or they're just too much of them, and how can our body adjust this condition?
Okay, if I understand that, could you repeat it slowly so that I can understand that?
Okay, let's just say, for example, E. coli is not the terrible bacteria to our body. Actually,
Yes.
It's quite necessary for us, but sometimes when you have too many E. coli, it's going to cause issues, and sometimes when you have not enough, it's also causing issue to your body. So how can our immune system know the problem is not happening with the bacteria is not bad, but it's just too much or too less?
Okay, so what happens here is that you remember that you have the symbiotic bacteria within the gut, and that's sometimes you're going to buy these they call supplements with bacteria and so forth to maintain a relatively higher number of the good guys, right? Versus
Yes.
so that if you have an invading bad guy, then they are outfitted. Now, what happens is that the good guys, so long as they're staying within the gut, okay, and in fact, there's a crosstalk, because you see, these bacteria are not just there digesting what they release molecules that are actually also being sensed by the immune system. There's a crosstalk between those two. What happens if there's an imbalance? You end up now having somebody with an upset stomach, diarrhea. So what happens is so long as those bacteria are staying within the gut and the microbiome, the microbial population is actually maintained, there's a balance, everything is okay. Now, As I mentioned earlier on, if they now get insight into the tissues where they're not supposed to be, they are going to be sent, they're going to be causing trouble. Why is that the case? Because some of them now get opened up and the content within the bacteria can end up causing inflammation because they are no longer intact. And of course, they can also replicate. Now to refer to your question, they get from there, it's going to be more nutrient rich. And they don't have competitor or they don't have any restrictions there. So they end up actually, the population ends up ballooning, and that causes a problem. So, Clostridium difficile, we make a lot of it.
Okay.
And what happens is that so long as it's within the gut, you're okay. How, when you go to the hospital and they want to check whether you're having a leak in your gut, what do they look for? Check your blood and look for the counts for Clostridium difficile. Because if the numbers are way high, they say this guy is there is a leak somewhere within the gut that the bacteria is doing what? Entering. Yeah. So I don't know. Did that answer your question?
Yeah, I think so.
It did? Okay. So yes, there is a mechanism by which the immune system or our bodies really sense these populations of these microbial population. And that's why some folks who have got unstable gut, they have these fecal implantation. Have you heard of that? Yeah. Yeah, you get fecal, and that's actually now the what? You're getting fecal material from somebody, from a healthy person, and then they get implant, and it works. It works very well. Yeah.
That's why you rely on them to be competitive against the bad guys.
Mm-hmm. Mm-hmm.
To maintain that balance.
Yeah, yeah.
It's pretty fascinating stuff, a whole new world.
Yes.
of immunology. Any other questions or comments, clarification, etc.? Okay, I think that's it. Until
all right, thank you. Let's see you on Wednesday. We wrap this up.
Thank you very much, Dr. Mwangi. Have a good evening.
Yeah, you too. Thanks.
Bye-bye.
Bye.